the question was, despite this stratification system we have to admit that many patients early relapse despite standard risk abnormalities, no functional high risk, no clinical high risk, but they early relapse. So we have to continue to understand the complex biology of myeloma to be able to identify all these high-risk patients from the diagnosis. And the single-cell technologies help us to appreciate the real nature of the disease. We know that this is a subclonal disease, but with single-cell technologies, we really see each cells, and we have access to DNA, RNA, epigenetic of each cells. So we have an overview of this heterogeneity, of this subclonal heterogeneity. And we recently observed that about 90% of patients display subclonal heterogeneity at diagnosis, with little subclones that are indetectable by a conventional method, but that we can detect by single-cell analysis. But more importantly, about 1/3 of patients display high-risk little subclones indetectable by routine practice. And we also showed, in particular with this little subclone displaying 1Q gain, that they seem to appear at relapse. In fact, they were already there at diagnosis, but we couldn't see them. But they have the same prognostic impact. So the identification of these little subclones from the diagnosis may help us to identify some high-risk patients currently identified as low or standard risk.